Hello and welcome to my URP presentation for 2021. My name is Andy Chia, and today I'm going to be presenting my project, Modeling Genetic Suppression of the POLE-E P286R human mutator cancer allele by intragenic mutations in yeast. So let me begin by telling you a bit about why we're doing this. There's this idea called the anti-mutator hypothesis, which was first proposed by Larry Loeb here at the University of Washington in the 1970s, and has laid the groundwork for a lot of interesting work in cancer. It bases itself on the fact that there are mutator phenotypes, or cells that have an abnormally high mutation rate, and the fact that at a certain point those mutations can be deleterious. And so the anti-mutator hypothesis states there must be a mutation burden where the selection of anti-mutator alleles, or alleles that attenuate or stop the mutator phenotype, is advantageous. Because otherwise, the only other possibility is that we experience or see error-induced extinction. And so this really becomes the best and only choice that we have for mutations. So in the HER lab, we mostly focus on understanding that relationship between mutation burdens and anti-mutator alleles. And in the past, we have relied on the workhorse, our yeast. And the reason why we do that is because yeast, mice, and bacteria all have relatively conserved, as you can tell from this alignment chart, conserved DNA repair mechanisms. And in fact, their sequences, when aligned, are very much the same. And what's more interesting is that in certain areas, we might actually see that mutations that cause deficiencies in DNA repair or otherwise cause mutation burdens to increase are actually mappable across different species. And so you can tell here, for instance, they're human, they're mice, and then there are yeast genomes that all have very, very similar exonuclease domains. And so when we see proofreading defects in human cancer, such as the 1 to 2% that cause colorectal cancer and 7% in endometrial, as well as others that can occur throughout different kinds of cancers, we can start to understand that perhaps yeast and other model organisms are a really great way of testing out whether or not we can identify different kinds of anti-mutator alleles. So let me just walk you through how we actually kind of go about that reasoning. So we want to look at the fitness of different kinds of yeast. So for instance, we have mutators that we've engineered that are haploids, which means that they only have one allele, or we have diploids that have two alleles. And so what we're able to do is figure out haploids have a mutation burden of around 10 to the minus 6 mutations per base pair, whereas diploids actually have a higher threshold because they potentially have two alleles. And so a lot of our work has been trying to identify what kind of EEC mutants, or again the error-induced extinction mutants, have what kinds of alleles. And depending on what kind of alleles they might have, we identify this region of volatility, which is essentially a region where they could potentially die out because of error-induced extinction, or they could potentially have anti-mutator alleles that arise. And so error-induced extinction, this is a bit of a visual for all of you, is very much understandable in yeast because they just cause yeast to die out and they can't grow anymore. So for instance, when we do have EECs, we can see in the colonies with the red circle around them, they grow these very small microcolony kind of phenotypes, whereas colonies or colonies that have not had EECs or error-induced extinction are able to grow relatively large. And so depending on what kind of phenotype they have, we're actually able to understand what kind of relationship each of these different mutants have. And so the mutator alleles all kind of exist within our DNA replication repair mechanism. So we might want to consider polymerase epsilon, for instance, or polymerase delta that are for the leading and lagging strand respectively. They're able to do proofreading through the exonuclease domain, or we might also want to consider our mismatch repair when we talk about different kinds of mutations that we might want to look for. So mutators and anti-mutators can change the sorts of polymerases that we might see. So my project is primarily dealing with polymerase epsilon. And so polymerase epsilon, which you can see over here, has a lot of different domains like the exonuclease domain, the palm domain, and other domains that we care about because each of these can be affected by a single point mutation. And so P301R, for instance, is a point mutation that occurs at the 301st of our polymerase epsilon. And so polymerase delta and polymerase epsilon both have a lot of points where we could have mutations that could actually be rather deleterious. And we can actually model poly cancer alleles because so many of our mutations that occur in yeast also occur in human cancer lines. And so we're actually able to take information from human cancers and tumors to actually model them in yeast. For instance, our discovery of spontaneous EEC mutants is because we can separate yeast into tetrads. So what we do first is get a diploid strain, which is heterozygous for both MSH2 and also a mutant form of POL. So both of these are mutant forms, and then we have a wild type as well. And so from there, we're able to sporulate and create tetrads. And as you can tell here, each of these haploid spores only have one allele of either the POL and the MSH. And depending on what kind of inheritance they have, a deleterious, as we saw earlier with the red, phenotype. After we get the wild types and cross them with the fastest growing colonies, we're able to create these diploids that, again, exhibit different kinds of phenotypes. And so our extragenic EECs, or mutations that occur outside of our gene of interest, cause a microcolony or a normal colony sized phenotype, whereas our intragenic EECs, or mutations that occur within the gene, have a normal phenotype. And so previous to my work, we actually identified quite a few potential antimutators. In our paper from 2020, my colleague Max Tracy was able to identify several candidate homologous to the cancer alleles that we see in a lot of different kinds of endometrial cancers that I mentioned earlier. And another thing that my project wanted to do was expand on that and try to find potential alleles in tumor cells that we haven't really considered before. So we went to the TCGA database and kind of combed through and found different kinds of alleles we might want to look for. So combining these leads, I was able to take both these cancer alleles and these mutations in our yeast have really high mutation burdens around the ballpark of 3 × 10⁻⁴, which is around the diploid mutation burden that we saw earlier, but also the fact that create new phenotypes when introduced with a mutator. So the mutator I used for this experiment was a very strong mutator. It's been identified in human cancers and is known as P301R. And because it's pol epsilon, it is analogous to the polymerase E P286R that we see in human cancers. So yeast transformation was the first step I did. So what I was able to do was transform yeast by adding a yeast vector in a bacterial genome, and then from there isolating and transforming it into yeast, and then growing them up on Lu2 plates as sort of a selectable marker in order to isolate yeast that had what I wanted, so the P301R allele. And from there, I'd pick colonies and then run a P301R genotyping gel. So this is a PCR assay where I run a PCR and eventually find out whether or not they were heterozygous. And I always wanted to find hets, which had these 2 bands. And from there, I would grow them on what we call synthetic complete plates, and then run a mutation rate assay. And so I would basically plate them onto synthetic complete for normal growth conditions, but also onto a non-synthetic complete plate. And from there, I was able to find out whether or not they had different kinds of mutations. And so here's some of my results. The mutation rates for yeast transform with P301R. Most of them are neutral, but there are some kind of surprising finds. For instance, the stars I have listed over here, and over here, and over here, they all kind of represent different kinds of alleles that might be anti-mutators. And we don't quite know yet, but it is quite surprising because some clonal isolates do have this phenotype and then others don't. And so our interpretation of that is that if they're in the cis configuration where we have P301R and a wild type, they might be collinear with an anti-mutator allele, which could lead to suppression. So we see that in some of these guys, for instance. However, we could also see cases where we have trans configurations where P301R hets are collinear with wild type, which means we probably have no suppression. However, we could also have P301R homozygous that collinear with anti-mutator alleles could have a sort of suppressed mutation rate of around 10⁻⁵. And so our next steps are trying to identify whether or not these are truly antimutators by genotyping them, but looking for these sorts of trends in human cancer. So one of our colleagues, Scott Kennedy, is working identifying different kinds of mutations that arise in tumor cells using duplex sequencing. So he would look for these mutations that our lab has kind of identified and find them potentially in single cells. And finally, I would like to acknowledge some people who helped make this project possible. My PI, Alan Herr, for being an amazing mentor and someone who's infinitely patient and wise, as well as my colleagues Annalie and Jason Lim for sticking it out with me.